All right, good morning all. I hope you're doing well this morning and the week is starting with a on a with a good mood. Can you hear me okay?
Yep, sounds good.
Awesome. Great. Are there any questions before I get started? Alrighty then.
How was your weekend?
My weekend was very relaxing. Since I cannot go anywhere because of the pandemic, I was just lounging and watching Netflix, South Park reruns, Rick and Morty reruns, and yeah, not much going on my end. So if you're not very active, don't feel bad. You're in the same boat. I'm in the same boat. I mean,
Good morning, Professor.
good morning, Vine.
Could you just go over the difference really quick between hypotonic and hypertonic?
Yes. So hypotonic. Hypotonic is the uh, hypotonic solution causes the water to rush into the cells because the surrounding content of water is higher than the content of water inside the cells. In the hypertonic solution, water rushes out of the cells because the content in the cell's water content in the cell's surroundings is lower compared to the water content inside the cells. Does that clear it up?
Yes, it does. Thank you.
No problem.
Think about this. GameStop
All right,
is
I'm
now
gonna mute
valued
Elisa here.
23.
Okay, I'm gonna start recording, share screen.
percent of their money. Other companies like AMC took advantage of the Reddit army.
Okay, so reminders. Quiz 2 is now open and you can take it anytime between now, or actually if you've taken it already, great, you're done for the week, quizzes wise. And the quiz 2 is due on Friday, February 5th. So it is due this week. However, you can take it anytime. You don't have to wait until last minute. You don't have to wait until Friday. And remember, the benefit of taking it before Friday is you could have take it once and then maybe something is not clear to you. You schedule office hours with me. I have office hours on Thursday and today, and ask me a question about what was not clear about the quiz. I will not give you an answer, but we can talk extensively about the concept and what are the differences between some concept and the other, and so on. So don't be shy to schedule office hours, and don't be shy. Ask me to clarify things during the office hours. That's what the office hours are for. Use this time. Use my time, and you're paying for this class, so um, take take whatever you can from it. And quiz 3 opens on February 6th, so technically it opens at midnight on Saturday, and you can take it anytime between February 6th and February 12th. It is due on Friday of next week. And we have a participation assignment for today. It is a short one. It has only 3 questions. It's mostly fill in the blank, or no, not fill in the blank, but choose a version of an answer from the drop-down menu. So I think it's going to be quite easy if you follow today's lecture. And today's lecture, it's going to be about DNA structure and gene function. So we've moved on to the next module in our class. We are done with the cells, with the energy of life, with the molecules of life and chemistry of life. And we are now moving into the new module dealing with the DNA and patterns of inheritance. So in the picture here, let me get my laser pointer, virtual laser pointer. In the picture here, you see a bacterial cell, and this thready stuff around it is the genetic material that has burst out of the cell. So technically, it's chromosomes, and the chromosomes are basically DNA wrapped around very tightly about some proteins. So this is essentially DNA, genetic material bursting around. DNA, excuse me, the genetics, which is the area of science that studies DNA and inheritance, is fascinating area of biology because DNA makes all processes within our bodies happen. So the function of DNA is to provide the recipes for proteins, and we have a lot of proteins within our body. Important proteins are enzymes, which speed up reactions, and without enzymes, nothing would happen in our bodies. Okay, think about, like, you probably are familiar with many applications of molecular biology and genetics, particularly the ones that are useful for us that we encounter every day is genetic modification of crops to increase the crop yield. So without genetic modification of crop, we would not have enough food to feed all the people on Earth. And it's been fascinating. It was pretty fascinating how the crops were genetically modified. So the crops were genetically modified to be more pest resistant. So that they were modified so that insects don't eat them as much. And the one I'm familiar with is inserting a gene from a Scorpio into a wheat DNA, and it caused the pests just not touch the wheat anymore because they didn't like the enzyme or the proteins that were encoded by that part of the Scorpio's DNA. Another fascinating example of genetic engineering is some bacteria have been genetically engineered to produce insulin. And insulin is a hormone within our bodies that helps us metabolize sugar or use sugar. People who are diabetic, for instance, they have trouble metabolizing sugar, and their blood sugar can get high, which can cause a suite of other problems. So there are two types of diabetics, the insulin dependent and insulin independent. So the insulin dependent diabetics, type 1 diabetics, they need insulin. They need to inject insulin in their bodies pretty frequently. So they monitor their blood sugar and inject insulin. And this insulin, where it comes from, it is produced by genetically modified bacteria that were kind of told to produce insulin on a biological level, of course. All right, so what is DNA? DNA is a molecule, and it is a type of nucleic acid. A little reminder about the molecules of life. There are four major groups of molecules that happen in biology, that occur in biology. The first group is carbohydrates. And the example of carbohydrates would be glucose, starch, cellulose. All of those are carbohydrates. And cellulose, remember, it's the same thing as fiber. The second group of molecules in biology are proteins, and the major example of proteins are enzymes. Also, our muscles consist of proteins. And two main proteins in muscles are actin and myosin, but we are going to touch on it when we study muscles, study the muscle system in our body. The third major type of molecules are lipids, and an example of lipid is, for instance, estrogen or testosterone. And the fourth type is nucleic acid. And apart from DNA being an example of nucleic acid, does anyone remember what other example of nucleic acid we've come across already?
RNA.
I did not hear you. Can you unmute and say again?
RNA.
RNA, yeah, and anything else? Was it an answer, Lisa Ann? Okay, that's hilarious. All right, RNA, great. All cells use DNA to store genetic information, which is what cells need to produce proteins. So you can see that DNA is a double helix, which has these, you can think of it as a ladder. And the rungs on the ladder are actually hydrogen bonds between nitrogenous bases of the molecules in the nucleic acids. So, and I will show what I mean in a couple of slides down. But for now, it's useful to visualize that DNA is wrapped around proteins. These little purple balls are proteins, and it's wrapped around into a chromosome. So DNA a chromosome is the combination of the DNA and proteins. So yeah, and this particular chromosome appears to be replicated already, and it will become clear to you why it's replicated when we go through mitosis and meiosis. All right, moving on. So DNA is composed of nucleotides. So nucleic acid is a general term for the organic molecule type. But DNA is considered to be a polymer, and the monomer of the polymer is called a nucleotide. So nucleotide is a building block of the DNA, okay? The three parts of the nucleotide are a phosphate group, depicted in yellow here, a 5-carbon sugar, or deoxyribose, and the nitrogenous base. And there are four types of nitrogenous bases in the DNA, which determine the letter that you're probably familiar with. It's ATGC, or adenine, guanine, cytosine, and thymine. All right, so DNA itself, what DNA stands for, it's named after the sugar, which is in the skeleton of the nucleotide. And the sugar is called deoxyribose. So DNA stands for deoxyribose nucleic acid. So Na is nucleic acid, and D stands for deoxyribose. Nucleotides form strands of DNA. So there are 2 chains of nucleotides here, okay? And there is one chain here, and the other chain is here. And the two chains are complementary to each other. So the chains on one side, they are connected with covalent bonds. So on the vertical axis here, these nucleotides are connected with covalent bonds, but the 2 complementary strands of the nucleotides are connected with each other with hydrogen bonds. So the dotted lines here are hydrogen bonds, whereas the solid lines are covalent bonds. So yeah, basically this slide summarizes that each DNA molecule is made up of 2 strands of nucleotides. The 2 strands wind together into a double helix shape, and this double helix shape happens because of the combination of hydrogen bonds and the covalent bonds. Like some of the molecules are attracted to each other, some of the atoms are attracted to each other because of the partial charges, which causes DNA to twist. And as I mentioned briefly, DNA strands are complementary to each other. The nitrogenous bases in one DNA strand form hydrogen bonds, or the dotted lines here, with nitrogenous bases in the other DNA strand, which holds the 2 strands together. And A always pairs with T, and C always pairs with G. So if you know the sequence or the order of nucleotides in one strand of DNA, you will be able to figure out the sequence or the order of nucleotides in the complementary strand of the DNA. So again, adenine always pairs with thymine, and cytosine always pairs with guanine. So any questions so far? I'm going to pause and let you speak for a while.
Yeah, I have a question. So the CGAT are connected to each other through the hydrogen bonds, and then the covalent bonds attach them to this outside part. What is the outside part again?
So the outside part, the yellow part is the phosphate group. The green part is the sugar, deoxyribose. Okay,
Okay.
so the deoxyribose is connected to the phosphate group, which is connected to the next sugar and the next nucleotide, which is then connected to the phosphate group, and so on. So covalent bonds are between phosphate groups and sugars of the next nucleotide, and hydrogen bonds are formed between the nitrogenous bases of the nucleotides in another strand of DNA, in the complementary strand of DNA.
Okay, thank you.
Okay, other questions? Tacos are completely great. That's how I learned it. Okay, great. Would you mind repeating the last part? From Melis, what last part are you talking about? Silence. Alrighty. Okay, so the important part is that DNA is a nucleic acid. It consists of the nucleotides. So the nucleotide is this one combination of the phosphate group, sugar, and nitrogenous base. The nucleotides are within one strand of the DNA, are connected through covalent bonds, phosphate to sugar bonds, and the two strands of the DNA are connected with each other with hydrogen bonds, and between the nitrogenous bases. And adenine is always pairs with guanine, and thymine always pairs with adenine. Okay, one more chat message. So, would you mind repeating the last part about nucleotides form strands of DNA? Yeah, so nucleotides, here's nucleotide number 1, here's nucleotide number 2, nucleotide number 3, nucleotide number 4. So you see that they are connected to each other. They form one strand of DNA, and they form one strand of DNA using the covalent bonds between the phosphate group and the sugar, deoxyribose. However, the DNA is a double-stranded molecule, and it is double-stranded because the nitrogenous bases of one strand of DNA and the other strand of DNA form hydrogen bonds, and that's how that two strands of DNA are held together. Does that clear it up? From Melis. Yay. Woo. Okay, moving on. So DNA packs tightly into chromosomes. And if the DNA were to unwind from the chromosomes and be just a double-stranded molecule within the cell, it would not fit inside the cell if it were unwound. So tightly winding around into a chromosome, or tightly winding around the proteins, helps it to be contained inside of a cell. And not only inside of the cell, it is contained inside of the nucleus. Remember, DNA never exits the nucleus. So this kind of helps it be neatly packed into the nucleus. So did I cover everything on the slide? I didn't cover genome. So an organism's genome is all of its genetic material in its cells. So all of the DNA within an organism is called the genome. So simple enough, maybe. So DNA, the function of the DNA is to provide recipe for protein production. So no lipids, no nothing, just proteins, okay? DNA contains the recipe for proteins. However, DNA does not exit the nucleus, but we know that proteins, well, they're not contained to the nucleus of the cell. So what happens is protein production happens in two phases, the transcription phase and translation phase. And during the transcription phase, the DNA, which contains the recipe for a protein, it provides the recipe to the RNA molecule, or RNA molecule, and the RNA molecule is then exits the nucleus and helps synthesize the protein by binding to a ribosome. So the process of copying the recipe for the protein from DNA to RNA is called transcription, and the process of actual synthesis of protein from the recipe contained in the RNA is called translation. So DNA and RNA molecules are similar, but different. So DNA is a double-stranded molecule, and RNA is generally a single-stranded molecule. So RNA is also a nucleic acid that participates in the protein synthesis. So one of the differences is the structure is that DNA is double-stranded and RNA is single-stranded. Among another differences between DNA and RNA is the sugar, which is a part of the nucleotide, which is a skeleton of the nucleotide. So in DNA, it's deoxyribose, whereas in RNA is oxyribose. Because of the difference in the sugar, as well as one of the nitrogenous bases, RNA is generally single-stranded, whereas DNA is double-stranded. So what are the differences in the nitrogenous bases? The three of the nitrogenous bases are the same between DNA and RNA. So adenine is present in RNA and the DNA. Guanine is present in the RNA, in the DNA and RNA. Cytosine, the same. Present in both DNA and RNA, but thymine, which is present in DNA, is not present in RNA. Instead of the thymine, RNA contains uracil. So the difference in the nitrogenous base and sugar is kind of the reason why RNA is not a double-stranded molecule like the DNA. Okay, I'm looking through questions, and can you please explain protein production again? Oh, I will go over it extensively, Mary, later in the slides. I'm not done with it. So difference between transcriptions and I will go over it again. Don't worry, I will go over it in detail next. Okay, and the functions. The difference between DNA and RNA are also functional. So DNA stores RNA and protein encoding information and transfers information to the next generation of cells, whereas RNA carries the protein encoding information and helps to make proteins and catalyzes some reactions. But the major function is to carry this recipe in the DNA to proteins and help make proteins. So the pairing, whenever RNA is synthesized from the DNA, you know what, I'll go over it later, but the pairing of the nitrogenous bases is summarized here. So whenever RNA is synthesized, if the nitrogenous base is called adenine, what is synthesized is uracil instead of, thymine, which is in DNA. So it's helpful to think about uracil as an analog, as an RNA analog in thymine. It's a very crude analogy, but whenever in the assignments, I ask you to give me a sequence of the nucleotides that would be synthesized from this recipe of the DNA, it's helpful to think of the complementary strands, and instead of thymine, which pairs with adenine in DNA, you can think about uracil as an analogy to thymine when I'm asking you to give a recipe for the sequence of nucleotides in the RNA. I hope it will become clearer later. It is we're kind of getting a bit of in the woods in biology, but I promise you it will get easier and it will not always be this difficult if it's becoming overwhelming for you, okay? So a gene is like a recipe in a cookbook. And what is a gene? A gene is an area in the DNA, is a portion of the DNA that provides a recipe for one unique protein. So a DNA molecule contains lots and lots and lots and lots of genes. And again, a gene is a portion of the DNA molecule, is a sequence of the nucleotides that encodes one specific protein. And the molecule of DNA consists of multiple genes that can provide instructions for for synthesis of multiple proteins. So the instructions for the protein synthesis are contained in the DNA. The genome determines which proteins cells can produce. The cell uses DNA to synthesize RNA, and this process is called transcription. And the RNA uses its genetic information to synthesize proteins, and this process is called translation. Now, I want to move to the analogy of transcription and translation. Imagine you're cooking something, okay? To cook, so you're cooking a brownie, you're cooking brownies, but you don't know the recipe for brownies. So you go to a cookbook or to a website and you decide to copy, you don't need the whole cookbook to cook brownies. Let's stick to the cookbook analogy, not the website analogy, because website would be more specific. So the cookbook contains like 600 pages. You don't need the whole cookbook to make brownies. So you open the cookbook to the page where the brownie recipe is contained. You copy the brownie recipe on the piece of paper, and then you read this piece of paper and you combine the ingredients for brownies and actually bake them. So the process of you copying the recipe from a cookbook is analogous to transcription. So in transcription, the RNA is synthesized, which is from DNA, which is basically the copying of the information, a recipe for a protein, into a piece of paper or RNA molecule. And when you are reading this recipe that was copied from a cookbook and actually combining all the ingredients together to make brownies, it is analogous to the process of translation. So what happens during translation is the RNA exits the nucleus, binds to the ribosome, and once it's bound to the ribosome, the protein is synthesized. So this is the reading of the recipe, and the combining of the ingredients happens in the ribosome when RNA binds to the ribosome and protein starts to be synthesized. So did that clarify the difference between transcription and translation? Are there any more questions at this point?
So the translation happens in the cytoplasm. Is that what I should write?
Exactly right. Translation happens in the cytoplasm and transcription happens in the nucleus. Very good observation.
Thank you.
Okay. Are there any more questions? Because I'm gonna move into more detailed description what happens during transcription and translation. So any questions about the analogy? Any question about DNA, RNA differences so far?
Is RNA also held together by hydrogen bonds?
Ooh, very good question. So the answer to that is no. Oh, come on. So remember, hydrogen bonds are formed between the nitrogenous bases in the DNA. But RNA is usually a single-stranded molecule. Mostly it's going to be a single-stranded molecule. So it's single-stranded because it doesn't form the hydrogen bonds. It is the nucleotides in the RNA are held together only by covalent bonds. And the reason why it doesn't form the hydrogen bonds, the reason why there is no complementary RNA strand bound to the other strand by hydrogen bonds, it lies in the differences between the sugar in the skeleton of the nucleotide, as well as the difference in the nitrogenous bases. In DNA, it's thymine, but thymine is not present in the RNA. Instead of thymine, RNA has uracil. So these subtle structural differences result in non-binding, like not forming double RNA strands. Did I answer your question, Joshua?
Yes, thank you.
Cool. Okay. So transcription. Transcription uses DNA to create RNA. Transcription is a set of chemical reactions that occur inside the nucleus, forming molecules of RNA. So again, transcription is the process of copying of the recipe for a protein onto a sheet of paper. And this sheet of paper in this analogy is the RNA. So technically, DNA creates this sheet of paper and copies the recipe onto it. So it's like opening a cookbook to a particular page and copying just the recipe for the dish you want to prepare. In transcription, DNA sequence, which is the specific order of the nucleotides, determines the RNA sequence. So to make RNA, base pairing takes place between RNA and DNA nucleotides. So if DNA, I'm not there yet, but what actually, should I go in there? I'm going to explain it in the next slide, but what actually synthesizes the RNA from the DNA is an enzyme called RNA polymerase. And whenever this RNA polymerase encounters the adenine, the nucleotide with the adenine nitrogenous base, it will synthesize the nucleotide with the uracil nitrogenous base. Whenever it comes across the cytosine nitrogenous base, it will synthesize the nucleotide with the guanine nitrogenous base, and so on. So it's helpful to think that DNA and RNA nucleotides, when RNA is synthesized, its nucleotides are synthesized in a complementary way to the DNA, to the sequence in the DNA. And in what way they're complementary is that adenine always pairs, well, it's not paired, but from uracil will be always synthesized from adenine. Guanine will always be synthesized from cytosine. Cytosine will always be synthesized from guanine. Well, it's kind of vice versa. And when RNA polymerase encounters the nucleotide with the thymine nitrogenous base, it will synthesize the nucleotide with adenine nitrogenous base. So in the next slide, there is a picture which might clarify what's going on a bit better. So the transcription that builds mRNA, we're getting more into the woods here. This RNA that is synthesized is called mRNA, and mRNA stands for messenger RNA. So it carries the message from the nucleus to the ribosome, and that's why it's called mRNA. Again, M stands for message. So transcription takes place in 3 steps. The first step is initiation, the next step is elongation, and the third step is termination. So during the initiation phase, the enzyme called, this purple blob here, this enzyme is called RNA polymerase. It approaches the DNA molecule and starts reading it. Okay, once it reads a sequence of the nucleotides called a promoter, it sends a signal to the RNA polymerase enzyme saying, oh, okay, it's time to unwind this double-stranded DNA molecule and start synthesizing the RNA, the messenger RNA. So the sequence of the nucleotides that send the message that it's time to start synthesis of the messenger RNA is called the promoter. So the second stage is called elongation stage. And during this what elongation stands for is the elongations of the RNA molecule, of the messenger RNA. So during the elongation stage, the RNA polymerase enzyme actually synthesizes the DNA. And here's the zoomed in image of this process. And the blue molecule here is the DNA. The green molecule is the newly synthesized messenger RNA or mRNA. And you can see that whenever the RNA polymerase encounters a nucleotide with the cytosine nitrogenous base, it will synthesize the nucleotides with the guanine nitrogenous base. Whenever it encounters the adenine, it will synthesize the uracil. Whenever it encounters the guanine, it will synthesize the cytosine, and so on. So let me put it in other terms. If the RNA polymerase encounters cytosine in the DNA strand, it will never synthesize, let's say, uracil. Okay, it will never synthesize adenine. It will always synthesize the nucleotide, RNA nucleotides, with the guanine nitrogenous base. And that is why it is complementary, and that is why it's possible to figure out the sequence of the RNA from the DNA strand from which it was synthesized, and kind of work backwards.
Professor, can you explain elongation again?
Elongation. Yes, actually, I will go over the steps in more detail in the next 3 slides. So it was just a brief overview of what's going on, and I will explain it again a couple of slides from now. Okay. So the termination is the final stage of the transcription, and whenever RNA polymerase, so RNA polymerase kind of reads the DNA, the recipe in the DNA, and once it comes across a particular sequence of the nucleotides in the DNA, which is called a terminator, it literally terminates the synthesis. So the terminator sequence signals that it's enough. It's enough of synthesizing of the RNA. Stop, and RNA polymerase stops and moves on to its next job. So next, I'm just going to reiterate, and that's for you to go over later. There's more notes on the following slides regarding each stage of the transcription, which will be helpful in preparation later. Okay, so during transcription initiation, RNA polymerase, which is an enzyme, and purple blob here, gets it all started. Once it encounters the sequence in the DNA called the promoter regions, which is a sequence of nucleotides that gets the reaction started or gets the process started, the RNA polymerase unwinds the DNA and starts synthesizing RNA. So elongation. During the transcription elongation, RNA polymerase synthesizes RNA. So this protein here, this enzyme here, it unwound the DNA, and it reads the DNA nucleotides, or rather from here to here. It reads the DNA nucleotides and synthesizes the complementary RNA nucleotides. So let's say RNA polymerase read that in this particular region here, there is a DNA nucleotide with a cytosine nitrogenous base. Okay, says RNA polymerase, I will synthesize RNA nucleotide with a complementary nitrogenous base. So the complementary nitrogenous base to cytosine is guanine, and that's what RNA synthesizes. And it will move on to the next nucleotide in the DNA. It reads the nucleotides in the DNA and understands that, oh, okay, so the nitrogenous base in the next nucleotide is adenine. So I will synthesize a nucleotide in the RNA with a complementary nitrogenous base, and complementary nitrogenous base in RNA to adenine is uracil, and that's what it's going to synthesize. So cytosine will always, the guanine will always be complementary to cytosine. Uracil will always be complementary to adenine. And what else here? ATGC. and thymine will always, okay, hold on, AUGC, I think it's all the pairs that are possible. So did that clarify what happens with during elongation? So do we have to know that it reads 5 to 3? No, just ignore that. It's not important here. Okay, great. So the final stage of the transcription process is the termination stage. So RNA polymerase reaches the end of the particular gene. And how does it know that it's the end of the particular gene? It's encoded by a terminator sequence. So what signals the start of the unique gene is the promoter sequence, and what signals the end of the particular gene is the terminator sequence of the sequence of nucleotides that signals to the RNA polymerase that it should stop the synthesis of the messenger RNA. Okay. So after the transcription termination, the RNA is complete. So at the terminator, or at the sequence of the nucleotides in the DNA that signal the termination of the RNA synthesis, RNA polymerase, it separates from the DNA strand, and it goes on to its next job. And after RNA polymerase leaves, the strands of the DNA fold into the double helix again, and they do it by themselves because remember, there are hydrogen bonds between the complementary nitrogenous bases that facilitate this process. So the hydrogen bonds form because one of the nitrogenous bases is slightly positively charged and the other one is slightly negatively charged. And because of this slight difference in charges, they will find each other and fold into the double helix again. Any questions about that? Any questions about what I went through so far, the transcription stages?
There's some questions about initiation, going back over those slides if we have time.
Yeah, we have time.
Okay.
So initiation. So everything happens in the nucleus, okay? The DNA is kind of wound up in a chromosome. Now, at some point, the enzyme, or this purple blob called RNA polymerase enzyme, will approach the DNA and it will start reading the sequences. It will start reading the sequences of nucleotides in the DNA. At some point, it will come across this promoter sequence, which signals the start of a gene, and the gene contains. Remember, the gene contains the recipe for a particular protein. So, RNA polymerase will once it reads the sequence the promoter sequence, it will start unwinding the DNA and move to the next stage, which is elongation. Did that clarify? So the person who asked about this was Ryan. Ryan, did that clarify? Yes, thank you. Ooh. Any other questions? So Jade asks the question, the point of this whole process is to make proteins, right? So far, the point of the transcription process is to make messenger RNA. We're not at the protein stage yet, we're at the messenger RNA. So the messenger RNA is analogous to a sheet of paper with one unique recipe instead of a cookbook. So DNA is a cookbook with thousands of recipes, millions of recipes, and our messenger RNA is a sheet of paper with just one copy of a recipe for a dish that you want to cook right now. Any other questions? Great. Okay, let's see. Okay, now to translation. During the translation, so translation again is analogous to actually cooking your dish, actually making those brownies from the sheet of paper that you've just copied the recipe to. So cells translate the mRNA or messenger RNA message into a sequence of amino acids. So proteins are a polymer, and the monomer of proteins are called amino acids. So if you have a sequence of amino acids, it can become a protein. And I just went over this analogy. So during translation, high level, what happens during translation is this messenger RNA that was just synthesized during the transcription stage. It exits the nucleus, binds to the ribosome, and the protein synthesis starts. The sequence of amino acids is generated. So all cells have the same genetic code. No matter which cells you take, it's always going to be the same type of nucleotides, the same 4 nucleotides, nothing new in any cell. So the 4 possible nucleotides can be the one containing adenine, the one containing cytosine, the one containing thymine, or the one containing guanine. And this, what this slide goes into is it introduces the concept of a codon. And the concept of a codon is a set of 3 nucleotides that encodes one unique amino acid. Okay, I will not ask you to memorize all of them. What I'm asking you to understand is basically that the sequence of the 3 nucleotides encode one unique amino acid. And amino acids are the building blocks of proteins. Okay. So what the codon is, the depiction of the codon, for instance, if the codon reads AAG, or this is the nucleotide with the adenine, this is the nucleotide with adenine again, and this is the nucleotide for the guanine, this codon will always be associated with the amino acid called lysine. This codon will always be associated with the amino acid called serine, and this codon will always be associated with the amino acid called valine. Again, I will not ask you to memorize like different amino acids that are associated with different codons. I just ask you to understand that what the codon is. And a codon is a set of 3 nucleotides that encode one amino acid. And where it becomes important in understanding what happens during translation. Because during translation, the codons will make sense. I will not go over the translation today because we only have 2 minutes left. But what I want to mention here, what I want to stop your attention at this part is I mentioned that amino acids are the building blocks of the proteins. But here in this slide, it's called a polypeptide. Now there is some difference between polypeptide and a protein. So technically, chemically, they are the same thing. Polypeptide is chemically the same thing as a protein. But protein, to become functional, a protein has to fold into a particular shape. And this folding will happen like not in the nucleus anymore. Well, the whole process of synthesis is not happening in the nucleus anymore. But remember when I went over the production of proteins in milk, the folding of the protein was done in the Golgi apparatus. So polypeptide, chemically, it's the same thing as protein, but we call protein a protein when a polypeptide is folded into a functional shape. Lauren, yes, in the Golgi apparatus is where the polypeptide is folded into the functional shape and is called a protein. All right, and that is 10:20 and the end of the class. Any last minute questions? Christine asked the question, did we go over everything for the participation? The answer to that is yes, we did. You may safely take the participation exercise now. Blaine asks, did we finish respiration? We did. Yes, we did finish respiration. I thought we did. Am I mistaken? No, we did. We did. So for our test on the 15th, is it due the 15th or does it open on the 15th? So test is on the 15th and it will be open during the class time. You will take it during the class time. Is it possible to upload PowerPoints before lecture? The answer to that is it's not my preference because I don't go over all of the slides before the lecture, and some slides I may omit because we will not get there, or I will realize it's not important really for this level of biology. So no, but if you go to the Zoom lectures, to the Zoom recordings of the lecture, the transcription of the lecture is posted along the video itself, and you can use it as notes. Moreover, you can search for key terms in the Zoom transcription, and it will point you to the definitions and the exact place in the video where the term is discussed. So I think it's even better than the notes. Anel, so polypeptides are folded proteins, vice versa. Proteins are folded polypeptides. Okay, well, thanks guys for joining in, and I hope you have a good week and see you all on Wednesday.